Good morning, everybody. If you can take your seats, we're about to get started. Got a lot of information to get through today, so we want to make sure we don't shortchange anybody. I'm Frank, and this is Elena. We're happy to have you here today for our biopharma panel. We have a slight change in lineup today, as all of you have seen in your programs for the insert. We're now going to be joined today by Birgit Schoeberl. Birgit is from Merrimack Pharmaceuticals, which is an innovative cancer research company. And Merrimack approaches, as they're based here in Boston, they approach cancer research from an engineer's perspective. So they build their teams encompassing experts across all different areas of business, healthcare, and engineering technology to really come to innovative solutions. In fact, just a few days ago, this integrated team approach brought them to their first FDA approval on Avide, which is indicated for treatment of metastatic pancreatic cancer. So great times at Merrimack.
Currently, Birgit is leading the discovery division at Merrimack Pharmaceuticals, where she focuses on developing therapeutic and diagnostic programs for oncology. She has been with Merrimack since 2003 in various research roles until becoming the head of discovery earlier this year. Please join us in welcoming Birgit to the stage.
Okay, thank you very much. I'm very excited to be here, and I hope the microphone works and you can hear me. Seems like it's working. And thank you very much to the organizers for the invitation, and I'm excited to be here. And I'm sorry that Sandra couldn't be here today due to a family emergency, but I'm trying to address sort of the same topics, sort of what are the challenges we face as an industry and what can we do about it, and specifically what is Merrimack doing about it. So the title of my talk is Cures Are Built, and you will see during the presentation why I chose this title and how we really use engineering principles to address some of the challenges we face as an industry. So the first part of my talk is to give you a bit of motivation of why we're trying to do something different and why we think that is necessary. So first, cancer. So Merrimack is focused on oncology. Cancer is a very complex system. It's not that you have only one cell type. You have multiple cell types. You have multiple lesions. It might be difficult to get drug to the tumor. As you know, you can give a treatment, but because of the plasticity of the tumor and the individual tumor cells, resistance emerges very easily. So how do we address these complex issues? Intuition alone, or just sort of very traditional approaches, haven't been too successful to date. So cancer is still a major problem. As you can see here, in 2012, we had about 14 million new cases of cancer, and it's estimated to double by 2013. So it's a large number of patients, and despite all the research we've been doing for the last 100 years almost, we haven't really made a big dent. But we have made some progress. So basically, since the 70s, where we only had less than 50% of the patients survive 5 years, now we have about 70% of patients and have an estimated overall survival of 5 years. So we have made some progress, but if you actually look at the adjusted death rates, here you can see in red when the war on cancer begins. For everybody who's seen the PBS show, is familiar with this term, you see basically not much has happened. Maybe now with immunotherapies, we see a little more of a difference, but even those don't work for every patient and don't provide cure for every patient. So something more radical has to happen to really make a difference and help patients. And just to give you an aspect, what that means actually for the industry. So part is that we're very inefficient. So if you look at oncology and the success rate from phase 1 to approval, it's only 7%. That's pretty inefficient. If you would basically build cars that way, nobody could ever drive a car here. So why is that? So we have a lot of candidates in early stage discovery and development. And then over time, they narrow down, and we get only to 4 to 6 approvals per year as an industry. So it's pretty amazing that actually we were one of the companies this year to achieve this milestone. But it's very difficult to get to this point. So how can we achieve maybe better success rates? And that was a question we as a company asked when we started to form Merrimack. So another interesting data point, and that is driven by the large number of early phase molecules that never go anywhere, is that actually over 70% of the cost of any approved therapeutic is spent in these early phases, even though any tox study, any preclinical study is actually pretty cheap to do compared to late-stage development. But because we do so many of them, it becomes pretty inefficient and expensive. So again, how can we make this funnel narrower and actually be much more successful early on to approval? So, and just to give you a sense of how we've been improving as an industry, it also looks pretty somber. So this is from a paper in Drug Discovery. If you look at the number of drugs, new molecular entities, per US dollars, or per billion US dollars of R&D spending, you can see over time we actually haven't improved, we do worse. So we really need to figure out ways to do better and maybe to do things a little differently. So our approach at Merrimack is to, one, organize the company slightly different, and I'll talk about that, and two, use engineering principles to understand the body and the cells as a system better, come up with design criteria for our drugs, and basically use these insights to drive our drug discovery and development. So if you look at how a signaling pathway looks like when you draw a scheme, it actually looks very similar to electric circuits. So there's no reason to try actually to use all the things we've learned in electrical engineering and apply them to biology. Now we have the tools to actually do quantitative biology and develop computational models that help us understand biology better, come up with specific design criteria, and engineer molecules that do what we want them to do, understand the biomarkers, and do much more streamlined drug discovery and development. So this is basically our workflow in a nutshell. So initially, we really want to understand signaling networks, cell-cell interactions, as well as the system's pharmacology, use that to set design criteria for our molecules, and I will show you examples of how we do that, engineer these to the specifications, and get them into the clinic alongside with companion diagnostics, where again, if you understand what your drug is doing, if you think you understand the biology in that context, you now have the ability to enter the clinic with pre-specified biomarkers, basically in your phase 1 trial. So then if you take this a step further, if you look at Merrimack and our pipeline, for the size of the company, it's actually fairly substantial. And the reason why we did this, or why we have developed all the molecules, and this is hard to see, I apologize, is to actually get to personalized treatment regimens. So the vision is, as a patient, you come to the hospital, your tumor gets analyzed, and you figure out what is driving the tumor, and then come up with the ideal combination of molecules, whether these are signaling inhibitors and chemotherapies combined, or chemotherapy and immune therapies, whatever it is, to help the patient. So that's basically where we want to get to, and we have built our pipeline to mix and match our molecules and come up with new treatment regimens. So we're very close to here. We're located in One Kendall Square. We were founded out of MIT and Harvard. We still have a lot of research collaborations here with BU, Harvard, MIT. We try to really stay close to academia. And we are a fully integrated company, which means we manufacture our early stage clinical drug at Merrimack for the biologics, and commercial and early stage for the nanoparticles. So we have 2 platforms. So very quickly, I want to give you a sense of what I talked to you at a high level, what that means actually in particular, and how we applied some of the systems engineering to our drug discovery. And then also give you a sense of what we are trying to do with the organization and sort of the organizational experiment behind Merrimack. So first I'd like to show you how we use systems biology and systems pharmacology to show you how we are technology agnostic, basically meaning we're not doing only antibodies, or we're not only doing nanoparticles. It's depending on what the biology tells us, that's the technology we apply for our drugs. And then introduce the concept of actually companion therapeutics rather than companion diagnostics. So the idea is you diagnose a patient, and then you figure out what is the drug or the drug combination the patient should be treated with, and then dive into the organization. So we are building a pipeline to address the hallmarks of cancer. So we believe it's important to not only do signaling inhibitors or chemotherapy or immune therapy. We believe you actually need to understand all of these 3 major aspects of cancer treatments and figure out how you combine them using systems biology and therapeutic engineering. So here's an example of how that could look like. So on the left, you see a biological network. So here's a signaling pathway of DNA damage repair. On the top, you see cell proliferation, very simple model on describing how cells divide and proliferate. On the bottom, you can see how the model can nicely describe experimental data. On the y-axis, you have number of cells over time. And you can use these models to actually predict what the combination does. So you can see here, this control grows, 1 inhibitor, chemotherapy, and then the combination, you actually get cell death. You can then do that for different chemotherapies and different nodes in this pathway. And you can see for doxorubicin, maybe this green inhibitor here, DNA PK, is a much better target compared to if SN38, another chemotherapy, would be your drug. So you can use this to A, identify targets, B, figure out combination treatments in a very rational way. Another example here would be how you can build now models that represent your body. So this is a pharmacokinetic model with the central compartment being your blood. And you can then figure out that for MM398, which is Onivyde, our recently approved molecule, it actually is important how long the drug is available. So it's a nanoparticle that slowly releases chemotherapy compared to the free chemotherapy. You can see with much less drug, you get better tumor growth inhibition than with the free irinotecan. This insight then defined how we dosed Onivyde in the phase 3 trial. And it's hard to prove, but I believe that's one of the reasons the trial actually succeeded. So just to give you another vignette, how the fit for purpose or fit for biology design works. So c-Met signaling, c-Met is a target that's been around for a while, has one problem. If you target it with a regular monoclonal antibody, you would activate tumor cells and they would grow instead of you see tumor inhibition. So Genentech was very smart and developed this one-armed antibody down here, which now doesn't activate, but is also not such a strong inhibitor. So from our modeling exercises, we know that avidity, so if you can anchor the molecule to 2 receptors, would be a lot more potent, and we came up with this construct up here, basically taking the one-armed antibody and adding on another piece so it can bind 2 receptors. So then you can use these simulations to basically show that in most tumor cell lines and tumor types, MM131, you would expect it to do better than METMAB. And then you can do the experiment, and you can see in red here, we actually are better in most cases, and where the model predicted we would be equal, we are equal as well. So you can see how the interaction between modeling and simulation can actually help you in the design of your therapies, help you also figure out where to go in which indications, and maybe how to combine your therapies. So the last application is basically to show you how you can use this to do some biomarker discovery and apply that to clinical trials. So preclinically, we observed for MM121, which is our anti-ERB3 antibody, that phosphorylated ERB3, the receptor the antibody binds to, is predictive of response. You can't measure the phosphorylation level in patient samples, so we needed a surrogate. We used our computational tools, identified biomarkers, and came up with basically the 5 markers that we wanted to measure in the clinic. It doesn't matter what they are. What you should take from this is you basically can, instead of measuring everything in your first clinical trial, you go into the clinic with a specified set of biomarkers that you're testing. And I show you now that this was actually really important for the drug to survive. So we went into 3 phase 2 trials in metastatic breast cancer, ovarian cancer, and lung cancer. In an unstratified population, you can see it looked pretty sad, control, and the treatment arms actually looked the same. So if you would get this data, you had no biomarker information, you would kill the drug because it doesn't do anything. Thankfully, we had that large biomarker program associated with all 3 studies. And basically what we learned is the following. So one, heregulin, which turned out to be the most predictive biomarker, is the ligand binding to the receptor, is prognostic. So what you can see here, if you just look at the control arms, so patients treated in this case, breast cancer, exemestane, paclitaxel, or olortinib, they all, if they were heregulin or biomarker positive, they progressed much, much faster than the heregulin negative patients. So this was a new insight that this particular growth factor is present actually makes patients resistant to standard of care. So if you now look at patients that are hereglin positive and the effect of 121, you can now see in all 3 cases, numbers are low, but you actually see now the effect of the drug and restoring sensitivity to standard of care. If you look at different indications, you can see that hereglin is actually very much abundant in all solid tumor types, and that 121 or anti-RB3 inhibition probably has a lot of potential, not only in these 3 tumor types. So that led us to still believe in the molecule and to keep developing it further. It now is in a lung cancer trial where we prospectively select patients. Just to give you a bit of a vision of where we would like to go and the idea of the companion therapeutics. So basically, what we are hoping to do at some point is that you profile the tumor, you figure out what is driving the tumor, and then you come up with different combination regimens for these patients. So just to give enough time for discussion, I want to give you a little bit of an idea of how we're thinking about the organization that basically keeps the best of biotech and also learns or applies the best of large pharma. So first, I said before, is we really want to be the experts in the hallmarks of cancer and being able to have therapies that address the different drivers, so whether these are chemotherapies, targeted therapies, or immune therapies. So how do we get to that, and how do we make sure the organization stays innovative? So we believe in the idea of bottom-up innovation, and basically the biotech company within the company have the entrepreneurs in the company. So on a regular basis, we have these innovation forums where the scientists within Merrimack propose new ideas. And you can see, based on how many people stand around a poster, how much discussion there is around an idea, maybe what is worthwhile pursuing, instead of management basically saying, this is what we should be doing or that. This way you get a lot of ownership. We then create these pods, and I'll talk a little bit more about this. These are very small research teams with the idea to fail fast. So take the idea, you are now an entrepreneur, you can show in 1 or 2 years that your idea actually is worth pursuing. And as it looks more and more promising, you get more resources, and the molecule or the team grows in a full-size team that moves to lead molecule and project team. So this is basically the vision for the pods, and with the idea to rapidly prototype molecules to, as I said, fail fast. And then on a larger scheme, so we basically inversed the traditional, more line-oriented organization. So we are organized by project team. Each of the project teams has a project leader. With the project team is a research team associated, medical director, clinical trial manager. They all report into these teams. And you can see discovery, the business unit or division I'm leading, is anything from idea to proof of concept. Once the molecule showed proof of concept in the clinic, it moves into development, where basically the focus is on great execution of registration trials and indication expansion. So the whole idea was that we can separate exploration and exploitation. So the area where we really want to get good at repeating the same over and over, to separate from where we want to be more innovative. And with that, I'll close and just give you an overview of our pipeline and where we are to date. Thank you. questions.
Yes, so if you want to raise your hand, so we'll bring a mic to you so that everybody else can hear.
Thank you. Great talk. I was interested, you showed early on sort of the cost of getting a successful compound through all the way through development to approval. Yeah. Did you go back and look at that in your experience and how you had been able to impact that graph for your company?
We are in the process of doing that. The problem with Onivyde is that it came through in-licensing from another company with who we merged 4 years ago. But if you just look, I think we have the most data to IND, and there we have been, so basically from now, or from the beginning to what's in the clinic, most of our molecules we moved through. So we have a better success rate up to today. The cost is also lower at this point. But the data, I think we need another 10 years to really have solid, solid data to compare. Any other questions?
Thank you. I'm on faculty here at Questrom. Yeah. You have a really interesting model of bottom-up innovation and an organization designed to support that. So that looks really interesting. I have one question because you talked about fail fast. Mm-hmm. How do you assure and how do you select which ideas fail? How do you assure that the better ideas proceed?
Yeah,
within a framework of discovery, not validation.
Right. Yeah. So for one, we do keep marketing and late stage out of these decisions. So we also have no committees. So we have a strict rule of having no committees to make these decisions. But we do have regular scientific reviews. So through scientific advisory boards with outside experts, also inside, and you can, if something is not going anywhere, you sort of see it, and the scientists themselves often see it. Maybe initially we're all trained to solve the problem and just keep going, but since nobody should fear about their job, right? There's so many ideas here, and basically what we're training or trying to build a culture is that people are not afraid to fail and to rather switch to a more promising idea faster. But it's not an easy process.
No, it's not easy, and thank you. Those are very specific mechanisms with people to address it. Appreciate your clarification.
Thank you.
Thank you very much for a very interesting presentation. One thing that I found intriguing was that concept of companion therapeutics. Mm-hmm. And I was wondering, could you comment on, is the current regulatory pathway conducive to bringing this concept into the clinic?
You mean for novel therapies or for just in general?
I guess maybe if the way I understood is that you're trying to combine, different molecules potentially early in development and, personalize them to patients. But how does that work in terms of the current regulatory environment where usually you just have one drug for a particular indication?
Yeah, so there are, so in form of clinical trials, you can run these trials. And if you want to take these to approval, you have to show that the combination is better than each drug alone. But it's new, and it's required to work closely with the FDA. I don't think there is an example where two non-approved drugs actually ever got approved together. I think one has to see how that shakes out. But you see a lot of these so-called bucket or umbrella trials done by either academia or across companies. You see more and more of that. And one reason is for a lot of molecules that require a genetic mutation to be effective, the incidence rate is often rare. So that means you have to screen, like if the prevalence is 5%, you have to screen so many patients and only 5% would be eligible for your trial. if you can provide another opportunity for the other 95, that's very much appreciated and more cost saving for everybody. So in our case, the prevalence of our biomarkers are, the incidence rate is higher, but it still is, if you prospectively select patients and you have to tell patients no, it's always better you have an option for them, especially if the procedure is invasive. So that's, I think, what motivates these trials.
Thank you for a very illuminating talk. One of your slides, you mentioned immunotherapy as one of the more promising treatment modalities. So I'm going to ask you to peer into the future. So when we invite you back in 10 years, what do you think will be with immunotherapy treatments? And the follow-up would be, how do you incorporate, let's say, metabolomic signaling with immunotherapy?
Yeah, so I think, so as we already see that even the PD-L1 inhibitors, there is a lot of hype. They work really well in some patients, but at the end, we also see only response rates of 20%. There's still a large fraction of patients who don't benefit. So I think the same principles, and we really need to understand how all the growth factor pathways, the immune pathways, how they all work together. So like in the early 2000s, all of us thought the targeted therapies of targeting EGFR would be the solution. They weren't. I think the immunotherapies show as single agents better effects to date, but I don't think it's the holy grail either. And I think really to conquer cancer, we need to figure out all the players to really get to a cure.
Hi, thank you for coming out and for the talk. I think you already spoke about this a little bit earlier as far as the decision-making process and what projects to move forward on. But I was also wondering if you could comment more generally about the interaction of science and business at your company and how they combine to get that idea from the lab to something that's deliverable. Specifically also like the role that MBAs, I guess, play.
No worries. We do like MBAs too at the company. So actually, if I go back here, so for example, in each division has like a mini CFO. So for example, I have my own CFO working with me, and he actually understands really well where each of these molecules are and like what are the milestones we need to achieve, where's the cost coming from, and he's part of the leadership team and is challenging also the project leads. So that's one example. Then as the molecules move more into the development, or MHS is our Merrimack Healthcare Solutions. It would be a whole separate presentation of how we try to set up the field model differently. That is very much an area where, MBAs are very much involved in marketing, setting up the field model, thinking about access. So I hope that answers your question.
Hi, thank you very much for your presentation. I really appreciated it. I have a question about the pods that you mentioned. Sort of how are those teams designed? How do they come to fruition? How big are they? And how does that span of control increase the efficiency that your company can work with?
Yeah, so we are a big believer of your own motivation. And so basically how the teams come into fruition is, so somebody presents at the Innovation Forum and is really passionate about this certain piece of biology, and it's something they really want to work on, and so they have the freedom to leave their team and basically start working on that. And we provide this flexibility. It might be that he or she has to wait a month or a quarter because they can't just go and take off from what they've been doing. The other model is that they have found other scientists who are already working in the pods to work on this program. So different ways, but in some ways, if you feel passionate and you want to be the father or mother of a molecule, that's instead of starting a new company elsewhere, you can do it within Merrimack. And then as either you're so persuasive and two others say, I really believe in this idea, I come and work with you, then they can go and do that. So it is somewhat self-organizing. And that's also how you can see where there's excitement or not. Like, if all the scientists decide to, I don't want to work on this anymore, I'd rather go and work over here with Paul or Emily, maybe that's not such a great molecule anymore to pursue. So it gives us a lot of data also.
Great. I think that is all the time that we have. Thank you, Birgit. I really appreciate it.
You're welcome. Thank you.